Thank you for joining us for our fall Outbreak webinar series. We are going to start with a slower overview of how to find data in the BV-BRC, and then over the next 2 weeks, we'll have a further look at our outbreak pages and our phylogenetics, and then our tools for analysis. So our outline for today is we're going to cover what the BV-BRC is, our data in response to the H5N1 outbreak in 2024, our data and outbreak response for MPXV, and then an overview on the website, workspace, and then a real focus of the topic. The time is looking at data searches in the BV-BRC. And hopefully there will be time for any questions, and feel free to stop me at any time. As I mentioned, we have the upcoming webinars next Thursday and then on the 17th as well. And we hope to see you at those. So if you're not familiar, the BV-BRC's mission is to provide a one-stop shop for data about human pathogenic and related bacteria and viruses into this scalable resource for comparative bioinformatics. And we support large-scale data mining, systems biology exploration, and use some machine learning to support the evolving needs of the infectious disease research community. And we hope to serve as a paradigm for research data resources. So if you find yourself wishing the BV-BRC had a tool or data type that we don't have right now, we are here to make this as useful for you. So please let us know so that it can be as useful as it can be. So some key features of the BV-BRC is that we support diverse bacterial and viral communities. If you study a pathogenic bacteria or virus, we have your data in our database, and if not, we can definitely add it. We have a unified data model and real-time data integration process, so we're updating data from GenBank on a nightly basis, as well as some of our other sources of data is continually updated to make the BV-BRC as useful as it can be. We support exponential growth of pathogen data. So a few years ago, we didn't have any SARS-CoV-2 genomes in our database, and now we have millions. So we are able to support the needs of, as data is generated, it will be easily accessible in the BV-BRC. We bring in data with consistent and accurate genome annotations so that it's searchable and useful for you to find and do this secondary analysis on exactly what you're looking for. So that also goes further into automated and manual curation of metadata, and we're always trying to make the metadata as useful as it can be for users. All of our services are run on high throughput computation, so you have the ability to do modular, interoperable, and compute-intensive analysis, all from your web browser. So that data is, those analysis jobs are easily run from anywhere in the world, and you shouldn't be slowed down by not having access to high throughput computing. Then we have integrative analysis of multiomics and system biology data using our visual analytical tools. And phylogenomic and epidemiological analysis for rapid outbreak response. We'll talk about that a little bit today, but that will be a main focus of our conversation on next Thursday. Then, as a BV-BRC user, you have access to a private user workspace for data analysis, sharing, and publishing. I'll go into what it means to have a workspace in a few slides, but this is really where you can put your data securely for you or anyone that you would like to see it, but we're never accessing that data, so that is private to you. Then if you are more familiar with computational analysis, we have programmatic and batch access via API and command line interface and an FTP, so feel free to use those to access any BV-BRC data. Then we offer outreach and education, like this conversation today. So our user community has over 50,000 registered users that come to the website about 25,000 plus times a month. We have over 20,000 citations, and our user base, I think, really reflects the audience that we have here today. So quite a diverse group, from basic infectious disease through clinical research, epidemiologists, bioinformaticians, data science, and more. Here's a quick overview of our services. So we support genomic services from assembly and annotation, BLAST, designing primers, variation analysis, phylogenomics, protein tools, multiple sequence alignments, metadata-driven statistical analysis. We just launched our docking service, which is a great one to check out, and we'll talk more about that in 2 Thursdays when we're looking at BV-BRC services. Then we have metagenomic tools like taxonomic classification, metagenomic binning and read mapping, transcriptomics, utilities, then some viral specific tools, specifically focusing on SARS-CoV-2 genome analysis and wastewater analysis. We support submission of influenza sequences to GenBank, so feel free to use that if you're submitting any sequences. Happy to help with that. I have an HA subtype conversion and our subspecies classification tool, which is accessible for both the viruses that we're talking about today, and then our outbreak tracking, which is currently focused on Mpox 2024, influenza H5N1 2024, and then SARS-CoV-2. You can just see a quick visualization of some of the data that and images you can generate with the BV-BRC. So translating over to the H5N1 outbreak, as I'm sure most of you are aware, influenza is a highly contagious respiratory illness caused by influenza viruses. And in 2020, there was a highly contagious H5N1 2.344b that emerged and spread all over the world, primarily through migrating birds. Then, as of 2024, the H5N1 strain was seen in both cattle and humans, which are new hosts. And so that prompted us to begin our outbreak tracking. And so over the course of the BV-BRC responding to this outbreak, we've shared relevant H5N1 news with the community, assisted in the submission of thousands of sequences for 2024 to GenBank. We generate phylogenetic trees and continue to update those, which you can see on our outbreak pages. Sent an infectious agent report in our newsletter. It's a great time for me to plug our newsletter. If you're not currently on the BV-BRC mailing list, I'd highly recommend doing that from our website, and I'll show you once we switch over to the website, but that's a great way to stay up to date with what's happening on the BV-BRC. And then we created the H5N1 outbreak pages. Next, my high-level overview for the MPXV outbreak is that is also an infectious disease that causes painful rash, enlarged lymph nodes, fever, headache, muscle ache, and back pain, and low energy, and it's caused by the monkeypox virus. And our outbreak response has focused on sharing this information out and just making sure that our users can have the genomic data that they need. But here you can see a map from our outbreak page that shows the detection of clade 1 and clade 2, and then also where there are publicly available sequences in the BV-BRC. So there are places in the world that have detection of clade 1 and clade 2, but the sequences either aren't publicly available yet or don't exist. So we're extra concerned about MPXV and clade 1 being seen outside of Central Africa in Thailand and Sweden. So our response is pretty similar to our H5N1 response, and that we're sharing relevant news with the community, generating phylogenetic trees that we continue to update, creating the outbreak pages, And we also added the MPXV subspecies classification tool to the BV-BRC, so that when you're under tools and services can now classify any of your sequences using any of your MPXV sequences using that tool. And all of the data in the BV-BRC is now classified by clade. So we will look at that when we're looking at the data. So I want to highlight next what a BV-BRC workspace looks like. So this will be a quick overview on a workspace, and it's really your personal dashboard within the BV-BRC, and that allows you to add information from your device and internally within the BV-BRC, and there are 3 options of personal, shared, and public, and that'll all make a little more sense when we're looking at the website, but just wanted to orient you with that. And the bulk of this conversation is on BV-BRC data searching. So to give you an overview on what our BV-BRC data is that we have over 11.8 million microbial and viral genomes. 11 million of those are viral, including over 8 million SARS-CoV-2 genomes, 1 million flu segments, and 32,000 bacteriophages. And we also have 10 eukaryotic host genomes to compare to as you need. Then, as viral sequences come into the BV-BRC, they're annotated across all genomes using bigger4, and that means we're left with 350 million genes, proteins, and mature peptides, and other genomic features labeled and ready for you to search. We curate the metadata, as I've mentioned, including host, collection date, geographic location, and other clinical and environmental metadata. We have over 220,000 experimentally characterized viral epitopes, 13,000 viral protein structures, 900 curated omics data sets, and 56 million microbial and host pathogens, protein-protein interactions, and over 1.6 million surveillance and 47,000 serological records. And all of this comes from public archives like GenBank, UniProt, PDB, IEDB, and from NIAID programs like SEERS, SysBio, functional and structural genomic programs. We also derive data from our internal pipelines and integrate that data, as well as there's plenty of other private data in your user workspace, but we don't have any information on what that is because that is private to you. And one data type that isn't listed on here is our sequence feature variant type data. I'm going to let Roshni talk a little bit about that, as that is relatively new. And if you were an IRD user, you might remember the sequence feature variant type data, but that is now on the BV-BRC ready for you to use. So some background on that includes, Roshni, are you ready?
Sounds good. So like as Anna mentioned, it's a sequence feature variant type. So basically it has 2 components, sequence feature and variant types. So what is a sequence feature? So sequence feature is a like stretch of amino acids which has some characteristics of interest. Like it may be a binding site, it may be an immune epitope, a structural motif, alpha helix, beta sheet, or something like that. So any characteristics of interest, and we are defining it at the protein level. And there are few properties, is like it's amino acid motif, which functional significance, I already mentioned that. With this, you can compare how conserved that section, that motif is compared to a reference strain, which we predefine for each of the strain or virus. We'll discuss it later. And then it can be linear, contiguous sequence or a discontinuous one. And sequence features, which in short we call it SF, can overlap and has no size limit. And our next one. So currently we bin sequence features, which we define into 4 main categories. One is functional, where the defined region has some functional property, like nuclear localization signal, its binding site, its active site, something like that. And then the next category is structural, that the defined region has some structural property. It forms an alpha helix, a beta sheet, or a barrel, anything like that. Then the third category is sequence alteration. Here, in publication or any other source, it's mentioned that a change in that position, in that motif, causes a functional change. Like, say, if I muted this residue, it affects the binding. If I muted that residue, it causes a temperature sensitivity or change in virulence or something like that. So that is a separate category. And then the last category is epitope. It's a coordinates for immune epitope. It can be linear, discontinuous, anything. So this is what the sequence feature mainly is. And now comes the variant type component. Now, what is variant type? So for each sequence feature, the extent of sequence variation observed within all the sequence that is publicly available is termed as variant types. So what we do, we take all relevant sequences for that protein in which the sequence feature is defined, and then do a multiple sequence alignment, and then observe the variation that is observed within that. So how it changes, whether it's conserved or not, and how the sequence vary. But we have few rules. So I remember I said we always define one reference based on the outbreak community, that that viral community. Like for influenza, we have defined each reference strain for each protein for which that we defined sequence features. For other viruses, we generally take the RefSeq sequence as the reference. And when we define the variant types, the amino acid sequence that is identical to that reference sequence is always defined as variant type 1. So that belongs to the variant type VT1 category. And the rest of the variant types, which is being defined, is allocated the variant type number based on the frequency, like how many proteins are there in that category. You will understand it more when we look at the data itself. And our next one, I think I have one. Yes. So this is an example of how a variant type looks like. So you'll have, there we have genome count, but in our database, that column is protein count. So we'll have a protein count and then a variant type number. And then you can see the amino acid position. Those are our reference coordinates, the coordinates of the region. So this sequence feature is from 137, amino acid position 137 to 147. And then we also have a column saying number of variations in compared to the VT1, so our reference. So this is how the variant type data looks. And you can see if there is something in the brackets, that means we have an insertion. If we have dashes, that means we have deletion compared to the reference. So, and apart from that, how can we use the SFVT data? So these are compiled from various sources. So it's just like you can identify the important amino acid motifs without knowing much about the virus or going deep into a publication or stuff. You can easily track variations within each motif or each sequence feature. And also, you can investigate the genotype, phenotype, association studies or statistical studies based on this data. So now I will do a demo from the BV-BRC, and I'll share my screen.
Okay, we can also wait until I get through the data, or you can do it now, whatever you're,
whatever, we can do it later also.
Okay, yeah, let's do it later so we can have people know where to find it. Sounds good. Okay. So when we're thinking about searching for data on the BV-BRC, there are really four main ways of getting information out of the BV-BRC. So the first way of searching will be right on the home page, and there's this big search bar, which I will show you in 1 minute when we get over to the BV-BRC. There's also our searches feature at the top, and you can search by organism, and we have all of the viral families listed we cover, and then featured viruses to easily get you to your influenza A genomes and your monkeypox virus genomes. And then after you leave the homepage, that search bar will go away, but there is a search bar that will pop up on the top of the page ready for you to search for anything you might be interested in. And all of these searches allow for filtering of data across the BV-BRC. So that will be a great place to start. And we're thinking about filtering data. This just lets us really generate the data set that we're interested in. So you can start with thousands of genomes or proteins and filter down to exactly what you're interested in. So now it's time to hop over to the BV-BRC. So here you can see our homepage. And so this is the view of the BV-BRC as a user. You can see that I'm signed in as myself. If I opened an incognito BV-BRC window, then you would see here to register or sign in. And so if you haven't registered with the BV-BRC account, this is what I would recommend doing. You'll have put in your email, username, name, and set a password. And there's also this link to register for our mailing list. That's how you get those newsletters. And I will put that in the chat so that you can all have access to that. So going back to the landing page for the BV-BRC, you will see a blue bar at the top that has our main things to look at. So as I mentioned, we have our organisms, bacterial pathogens and viral families, and then the featured viruses. And each of these, if you click on them, will take you to that overview data page, which I will do in a second. Then we have our advanced searches, which you can search by genomes, proteins, surveillance data, sequence feature variant types. This is really to get you to search for what you're interested in. Then that tools and services menu. And so this is how you're going to run an analysis in the BV-BRC. So you just click on one of those and it would take you to the tool page. Then you have your workspaces. And so when you make a BV-BRC account, you will get a workspace with a home directory, and then that will have 3 folders that are automatically generated. Those are your genome group, feature group, and experimental group. We have public workspaces that have some good information and are able to be shared with the public, but even better are our workshop folders, and these will have all of our week-long workshops that we host, all of the training material that is associated with those. And so that would be great if you're a professor who's teaching with the BV-BRC, or just interested in a longer scale how-to guide for the BV-BRC. Then you can access your jobs from up here, as well as down at the bottom right-hand corner. You can see how many jobs you've run and if you have any running, and then any of your genomes that you've added yourself. We have our help documentation website, and our news and announcements and reports and analysis are found here. And this has really got some good resources to get comfortable with the BV-BRC. Then our about us and contacting us. And if you ever have any questions under about us, you can hit contact us, and that will pull up a contact us form, and that will get sent, and someone from our team will answer that. If you haven't logged into the BV-BRC in a while, you might see this Outbreak section as something new, and so this will pull you right over to our Outbreak pages that we're going to be talking about next week. And then we have our news and announcements section, so highlighting that we were renewed just in September, so we're excited to keep bringing you the BV-BRC, but you can flip through these to see anything else that's happening on the BV-BRC. And then our LinkedIn feed is here with our most recent posts, so that's a great way to keep up with anything that's new happening on the BV-BRC. Then here we have our main search bar that I mentioned. So you can search for anything that you might be interested in, actually hemagglutinin, so you could search that from here. That will be a very similar search to this one at the top bar. And that will say that, unless I spelled it wrong, which I might have done, it would do a better job of pulling it up. That was my bad. But let's go into our organism search. This, I think, is a really good way to orient yourself with the data that you're interested in. So if we're starting from an H5N1 perspective, let's think about our influenza A virus as a starting point. So you click on that under featured viruses, and that will bring you to the taxon view and the overview page for influenza A virus. So I've got a lot of information here. So we'll just go through that quickly. So at the top of the taxon view, this is a really good way to check in to make sure that you are looking at all of the data that we have about something. So we're starting all the way from viruses down to influenza A. And so we have our 1.1 million genomes that are all of the influenza A segments that we have. Then reference genomes that exist, some metadata features, and any recent PubMed articles that relate to that. The next tab is our taxonomy tab. So here you could go to the specific subtype that you're interested in, and which we can do. So if we go down to H5N1, I'm just going to zoom back out so that you can see this green bar. So for this serotype, we have 30,000 genomes. And again, think about that as 30,000 segments. And then we can go to the genomes view. And then that will just be those H5N1 subtypes. But let me go back, and this would be a fine place to start filtering from. I personally like filtering from keeping this influenza A virus at the top, and this would just be a good place to check in on like what you're actually looking at. So we also have a strain tab, and so there are no filters here. So for these strains, we have ones that have all of the segments. So then you can download that data or use it for analysis as you see fit. Going further to the Genomes tab, which is where we'll spend most of our time, this page automatically opens with our genome name, strain, GenBank accession, size, collection year, isolation country, hostname columns opened, but you can add other columns as you would like. So to do so, you might be interested in seeing H5 or H type. So, and then we could also add N type. And then this is really just whatever is helpful for you to visualize what you're interested in. So all of these different data attributes can be added as you need them to be. So you can scroll down and add as many as you would like. But then the other strong feature here is to filter. So this black bar will pop up, and you can also change what filters you'll see. So you might be interested in filtering on H5 clade, so that doesn't automatically come up. This is scrollable, so I'm on a Mac holding with 2 fingers, and you can also scroll this back and forth, and then you can also make it bigger or smaller to see stuff that might be hidden if you want to see more. And the columns can also change their size, which will allow you to have more columns open. And then I just clicked on that instead of just moving it. So now it's sorted by the CDS column. You can sort by collection year. And so right now we still only have influenza A virus. That's as specific as we've gotten. We can then filter by subtype or H type. Either way is totally fine. If we do H type, let's do H5. And so this number down here at the bottom is now 100,000, but we still see 1.1 million up here. And that's because we haven't applied this filter. So that's why I'm stressing to keep an eye up here, because once you apply that filter, that will say only influenza A viruses that have an H type of 5. So we'll also do the N type 1, and I will apply that filter. So you'll see all of the other H and N types went away, and the subtype is H5N1. Just go here to see that, then see that there. So for the purposes of filtering this data, we'll also go to 2024. I'll apply that filter. So I scroll down to collection year, hit 2024, then hit apply. You can see that we have sequences from across these different countries, all these states in the US, different geographic groups, different host common names. So you could really begin to compare this however you were interested in. And different host groups, primarily avian, which makes sense. And then primarily 2.344b, which also makes sense. But you can Then also filter by segment. So if we were interested in a hemagglutinin comparison, maybe we want to only have segment 4. So that means that then these are all just segment 4. And then you could begin to really do some sort of comparison with those segments. When you click on a specific row, you will get, well, first you'll see that there are 1 out of 200, and then 1 through 200 out of 2,243. So that means we have 2,243 H5N1 segment 4s from 2024. You could switch to a different page, and that each page only has 200, but you can flip through all of the pages. If you click on one specific row, that will fill our green bar on the side with additional information. And so can have a quick reference guide for our data types, can download that one specific row. You can go to the genome view for this one or put it in a group. And so when we're going to put it in a group, this is how you can begin to craft your analysis. So if I already had an existing group that I wanted to, this turkey sequence was very important to me to add to that group, I could add it to an existing group. But since this is the first time we're looking at this, I'm going to put it in a new group. It defaults to your genome groups, but feel free to organize your folders as you see fit. But then you can label the group as you wish. And then you will add that sequence to your group. And you'll get a message saying that that group was created. Now I might also want to add some of the neighboring sequences. You can select them individually. You can select everything up here with this little arrow. And then add those to your group. So then you can search by whatever you want to, and then that'll put this in that group that I just made. And the most recent genome group will always be at the top, so it should be pretty easy to add things if you're working on something at the same time. So now it's adding those 6 items as you see in the bottom. And since I deselected the one that I originally had, it was 6 unique, but it won't duplicate when it adds genomes to your group. So going back to having one selected, my computer isn't loving having all this going on, but 1 second, we should have just one selected. You will see kind of an overview of the metadata associated with that segment. So this one is 1,704 base pairs. This is the taxon information. It's a partial sequence. Links out to bioprojects and GenBank accessions. How it was sequenced. GC content. Any information that we might have on isolation source, host information, and when it was inserted into the BV-BRC. And then, so all that information is there. I'm gonna then just make sure that we're only looking at clade 2344B, so applying that other feature. And that is an annotation that's added by the BV-BRC subspecies classification pipeline. So if you look at adding a column for H5 clade. You'll see that those are 2344B, and that's something if you have a sequence and you'd like to get that classification, just hop over to our subspecies classification tool, which we'll talk about in 2 weeks. One important thing to note is that these filtering of the data set based on metadata features is all from the genome column. So this genome tab is where you want to get all of your filters set. So I will also add a North America filter. And so then these are only H5N1 sequences from 2024, segment 4, 2344B in North America. And then the subsequent tabs are all based on that specific, all of these extra attributes, because you can't go in from the feature tab and change the collection year or the geographic group or the host. So that's just something to keep in mind, that even if you're interested in looking at features and proteins, it's important to get all of your sample metadata filters taken care of from the genome tab, which is a question we get sometimes. So as you move over to the features tab, you'll see that these metadata features are applied. The features are all from our Vigor annotations, and that has, for those 1,800 genomes, we have 8,000 features. And so here would be a great way to filter for a specific, oops, sorry about that, a specific feature of interest. So you could add to look for HA1 or signaling peptides or hemagglutinin. And had I spelled it right earlier, this is where we would have been brought to. But so for those 1,800 genomes, 1829 and 1795 have a hemagglutinin product. So then you can then make a features group of all of these 1835, or get the FASTA data, prompting for multiple sequence alignment, or putting those in their own groups. So we have that automatic feature group file. And then you can create a group to do further analysis. I just saw Deborah's question about preferred browser. We primarily use Chrome and Safari, but it should work on any browser. Going back to our features, you can see that there are some different metadata fields associated with that, and you can also then add features as you would like, and also add different filter systems. So both of these have that flexibility like we did on the genome side. The same thing translates over to proteins. So this will be the amino acid sequence instead of the genomic sequence. And you can then find that same product field and sort as you see. So we have our 7,000 for our 1,800. Then you could hit whatever product you were interested in and then make a protein group to do further analysis on the BV-BRC or download or use as you see fit. We also have protein structure data that comes from primarily PDB, but so can see what institution it comes from, what method, the structures, specific genes. You can search here by keywords, which you could also do on any tab. So if we were interested in HA, then that'll pull anything that has HA. From a, and this is influenza A specific, so not going to be structures for this specific group, but you can now have a new button on our green tab, which is our structure view, which will then take you into a protein structure view. And you can, once this loads, see this metadata about the structure, the sequence at the top, and then the structure will come up. And you can move it around as you would like, color on different things, delete the waters, select and zoom in onto specific things, and a whole bunch of different things that you can change in here. And then you can also take a screenshot or highlight different things. So a whole bunch of flexibility in the protein structure view and on the BV-BRC, and clickable from these structures. See at the bottom, as you click on things, there'll be information here. We have our domains and motifs, epitopes. See these different epitope information. And the great thing, I know we've spent a long time talking about influenza A, but this whole method and structure is the same for whatever virus you're looking at. So this is super applicable for whatever virus you're looking at. So the other virus we're interested in, the monkeypox virus, we have the same set of tools and things to review. The epitopes come from different assays, and you can review those here. We have our experiments tab, which has some experiments that have been done with this sort of sequence. Then our surveillance data. This will show you all of the influenza A surveillance. Then that can take you to our surveillance map, which will show you another view. So that was just one specific point, but you can take all of the, we can filter here for, if we did the year 2023, and this is from our SEER collaborator, so thank you for all the work that you do if you're a SEER person. And then, so what I just did was took collection year 2023 and selected all of the surveillance data we have for that. And then that will generate a map where that data is. All of it is in the US or in Canada, so that's why it landed there, but it will land depending on that. You can add highlighting the flyways. And then you can click on a specific site and see what the flu positive rate was. And you can partition by different time points and change any colors as you see fit. And then I will just quickly mentioning our serology tab, which also has some good data. And then I will let Roshni talk about the sequence feature variant type data, and she can share.
Yeah, I'll share my screen. Can you see my screen, Anna?
Yes.
Okay, sounds good. So as Anna showed, there are two ways to access the sequence feature data is like one from the organism tab. So currently we only have sequence feature variant type data for influenza A, but we are planning to extend it to other virus families. So let's take influenza A as example. So we went to the featured virus page, and then from there we have the sequence feature variant type tab. And then you can see here is a list of all the sequence feature that are being defined for influenza A. We have around like 5,000, more than 5,000. And whatever functionality that Anna showed applies here too. So you have filters based on that filter. You can filter this table. Say you want only the H1s. We select the H1 and then you can see these are all the H1 sequence features. So this exactly follows what Anna talked so far. What I will do is we'll come back to the same data, but from the in a different way. So we'll do from the searches. So in the search tab, under the advanced search, we have sequence feature variant types. We click on that, and this is our advanced search page. So in the pathogen group, currently we only have influenza A. As more viruses become available, it will be available in this drop-down menu. You can select that, and that will customize your this search area. So you can see, like from the beginning, you can filter down based on the segment, protein, based on the feature type. And if you select HA, it opens up more, like you can select subtype, specific subtype you're interested in or not. Like if you don't select anything here, then it gives you all the HA SFs, sequence features. So let's select H5 and we hit search. We have similar for NA2 if you want to do anything, but let's just take one example. And so we have HA, H5, and let's search it. And we have the sequence features which belong to the HA protein and which were defined for the H5 subtype. So we see we have 587 of those. If you look at the name, it tells you that which subtype or which protein it is defined in, what virus it is defined in, and also gives a brief description of what type of what effect that sequence feature has. So you can do keyword search generally. Here also, like based on if you even filter from the search, the advanced search menu, still the filter option here works. You see it has already applied the HA and H5. You can further filter down based on other categories, and you can add more if available there. Now, if you select one, say this one, And once you select one sequence feature, it will give you an option to look at its variant types. And before that, on this side, like when you select a sequence feature, you see more details regarding that sequence feature comes up. It gives you the source strain where it was defined, publication ID from where it was defined, and comments like what's the effect of that sequence feature. And you select one sequence feature to look at the variant types. You click on this tab. It takes you to the variant type table. See, I was talking about we have the protein count here, and then the variant type, what membership, the VT1 is always the reference. And the total variation column gives you how many changes are observed compared to the reference. You can find for a particular variation, or if you want, say, you want to look for where the 513 position has R, and you search it, and it gives you everything that has R in the 513 position. What these symbols mean, we have a index here at the end. You can see dot means it matches whatever is in the reference. And then if we have a bracket, it means insertion relative to the VT1 dash insertion, deletion relative to the VT1 and such. And if you click on this number, say you are interested to know what sequences lie within the VD7, you click on this 61, and it opens a feature group, like Anna said previously, which has the accession of the sequence and the genome name, like from where you can get the strain name, and for whatever that 61, what those 61 sequences are. And you can select from here everything, and you can group and save those for later for your other analysis and stuff. So this is how it works. The search column has, like advanced search has another feature, which is the sequence search for the sequence. Here, if you are, say, if you are interested in a particular sequence in a motif, say you're looking for ABCD, and you want to look which variant type has like those 4 amino acids together, then you can use this. You can click on this information, and it will give you how to search. Say if you want to search for insertion, you use brackets and search for it, and how to do it, how to do things. Like if you're looking for exact match, you want to look for sequence which starts with the RER, then you do RER, then asterisk sign. So it will give you the guide how to do it. If it's not too specific, say you search for RE, and it's not very specific, right? And then if you search for it, for now it's giving. Sometimes it may not give you any answers because there are too many hits. For that, it will give you a warning, say, please make it more distinct or more specific. So just for example, let's do we do anywhere star R star RERR star. We are looking for this motif, and if we search it, say 54 of the sequence feature have that in somewhere in its variant types. And let's check one and see you have the RERR here. So you can search based on the amino acid sequence pattern 2. So that's it for me.